Shall I start? Okay, thank you, sir. Hello, good evening, everyone. I'm Dr. Ashish Chaudhary, and today we'll be talking about one of the deadliest diseases, that is the rabies. And we'll be talking about some of its vaccine and one of the latest vaccines by the Cadila, which is a recombinant G protein vaccine, and are very much a new baby in the market and a new product portfolio, and comparatively a better vaccine. And I will be talking about that. And I'll be just sharing some knowledge of mine on the rabies disease as well. So let us talk. So we'll be talking about the rabies disease. We'll be talking about the animal bites incidences in India, about the epidemiology of the rabies, how the vaccines have evolutionized since 1885, what are the current challenges and what are the unmet needs. Then we'll be talking about a bit of introduction of this new novel vaccine, this technology, as well as some clinical studies on that, and finally the takeaways. So first of all, the rabies disease. Rabies is definitely, it is a zoonotic disease which is being spread by the animals. It can be either dogs, cats, bats, foxes, and it is caused by the rabies virus. And this virus is enveloped in a 5 structural proteins. That is the N protein, P protein, M protein, L protein, and the G protein. Out of which the G protein is the major antigen in the formation of the neutralizing antibodies against the rabies antibody virus. So about the incidence and the epidemiology, globally the incidence is about 59,000 rabies deaths per year, which is quite high. Just hold on, excuse me. Hello. Sure. Okay. So globally, the incidence is about 59,000 rabies deaths per year, and out of which in India, it is about 20,000 rabies deaths per year. If we differentiate it into the rural as well as the urban population, the maximum contribution is from the rural side. That is about 90% of the deaths, they occur in the rural areas. Just hold on. So, is rabies fatal? Definitely it is 100% fatal, but it can be, we can definitely prevent it through the vaccination. So how did the rabies vaccine evolve? Everyone knows in about 1885, Louis Pasteur was the first scientist to invent the rabies vaccine. It was through the nervous tissue. It was a live attenuated vaccine which was derived from the spinal cord. It was a non-standardized dry preparation, and about 31 dosages were required to increase the immunogenicity at that time. In 1911, again, inactivated vaccine was developed from the nervous tissue itself. It was a minimal neural content liquid preparation in which 14 injections of 0.5 ml per day were given for 14 days. Finally, in 1976, the cell culture vaccine came into the existence. It was an inactivated vaccine. Standardized lyophilized immunogenicity was seen, in which 5 injections were given about with a 5-dose regime on 0 day, 3rd day, 7th day, 14th day, and the 28th day. So what are the current challenges in this new cell culture vaccine, which was developed about 50 years back? So the current rabies vaccine is a cell culture vaccine. It is an inactivated vaccine and about 50 years old technology. There is some inconsistency in its potency. The stability of the vaccine, definitely, it is to be refrigerated, hence it is highly unstable. the supply is short and some adverse events are also seen. Decreased compliance is beyond 3 injection is the major issue of this vaccine. So current challenges. So ultimately the current challenges is the non-compliance beyond the 3 injection is the major issue. So finally, the introduction of a new vaccine meeting the unmet this medical need with a simple and a shorter regime. So this new vaccine has been introduced, which is the first protein subunit vaccine prepared by VLP protein platform technology. This is the virus-like platform technology. First vaccine with 3-dose intramuscular regime, that is the 0, 3, and 7 days. Excuse me. It has a better compliance due to easy flow of the regime and as well as the less visits, and finally convenient to use because no reconstitution is needed. So what is this virus-like protein technology? It is basically the native virus and the virus-like proteins, which works as vaccines. So what we have talked that the G protein is the major protein for the immunogenicity, but it is difficult to stabilize. Hence, the G protein is isolated using the virus-like protein technology. Virus-like particles are the protein complexes which exist as nanoparticles and are similar to that of the viral genome. But they do not contain the viral genome. They cannot replicate, hence they cannot be pathogenic, but they can mimic the viral antigenicity. They are capable of inducing a strong humoral as well as the cellular immune response. Hence, they work as very good vaccines with lesser side effects. This technology has also been published in the Association of Prevention and Control of Rabies journal in India. So some clinical phase trials which have come out for this vaccine, first of all, the seroprotective data from the day 14 to day 270, which was compared with the other competitor vaccine, and it showed the same seroprotection data as same as that of the competitor vaccine. So clinical study about the phase 3 trials where the safety and the immunogenicity of this recombinant nano rabies G vaccine was administered as simulated post-exposure immunization to evaluate the immunogenicity and the specificity of the rabies G protein vaccine administered as the simulated post-exposure immunization in healthy volunteers. 800 subjects were taken, and the primary objective was the seroprotection at the 14 days post first dose, and the secondary objective was the seroprotection at the day 42 post first dose of the test vaccine, and safety by monitoring the subjects with the adverse events. So the results were consistent across the different age groups and the genders. Non-inferiority for seroprotection, primary endpoint at day 14 and day 42 were met. Tests and comparator are identical in seroprotection rate on both 14 days and the 42 days. And the new vaccine had shown a better safety profile as compared to that of the reference vaccine. So with this, I would just like to conclude that this new vaccine, the rabies, was non-inferior to the reference vaccine. It had lesser side effects and adverse effects compared to the competitor vaccine. And the beauty of this product was the 3-dose regime compared to the 5-dose regime of the cell culture vaccine, and a better compliance to the patient. And finally, there were some acknowledgements from Indian pharma world as well as from abroad to this vaccine. With this, I would just like to conclude off my topic. Thank you so much for your patient listening.
Thank you so much, Dr. Ashish, for the informative talk. We truly appreciate your efforts. Last but not the least, we would like to inform you that rabies has won 2 prestigious awards in innovative category, like India Pharma Award and Golden Globe Tiger Award. So thank you so much, Dr. Ashish. Thank you.